centigrade, so no life there. Venus is not quite as hot, but still too close to the sun. And then you have Pluto. We saw the New Horizons give us some really nice, beautiful images for Pluto. Much too cold, much too far from the sun. Then you're left with that one right there, and that one is Mars. And that's actually a pretty good place to look for life. This is what Mars looks like now, but this is what Mars used to look like. It used to be a watery planet. What Mars had is very much like what we have. It had an iron core in the center of the planet that would spin around and create a magnetic field. And that magnetic field was like a big balloon around the planet that deflected the solar wind. When that core stopped spinning, you lost that big magnetic cloud, that big balloon of protection around the planet. And the solar wind hit into the planet and blew the atmosphere away. And when it blew the atmosphere away, it blew the water away. That doesn't mean to say there's no life now. We have a lot of evidence that there is water under the surface on Mars. So it's possible that if there is life on Mars, it's retreated to the subsurface where the water is locked away and can't be blown away by the solar wind. You also need that because if there's no atmosphere, all that radiation from the sun is hitting the surface of the planet. And when it hits the surface of the planet, it creates a lot of damage. You heard about the ozone hole in our planet. If you go down to Antarctica, you have to be very careful with sunscreen because the radiation from the sun is very damaging. The whole planet is like that. In fact, the surface is very inhospitable. It's cold and it's constantly bathed in radiation that would kill life. So if life is going to be there, it's going to need to be underneath the surface. Now, this gives us an N of 2, where we can look for the potential life. So we've got Earth, and now we've got Mars. But there's actually a lot of other places in our solar system we can look. And that's because there are other planetary bodies, so these are not planets, but other planetary bodies that potentially can house life. So there's over 200 bodies in our solar system that are big enough that they kind of span around and made planet a round thing. And most of these are moons, so these are not to scale. These moons are different sizes, but this is our moon here. This is Enceladus. This is Jupiter. Titan is Jupiter. Europa is Saturn. And these also have the potential for life, because what you need for life is water. So this is Europa. It's an icy moon. It's a dirty snowball. You're looking at the surface of a dirty snowball there. When you kind of zoom in and look at the dirty snowball, you can see it's crisscrossed with these cracks, like ice cracking on the surface of the pond. And you also kind of get these blobs, like pieces of ice that have cracked below and floated to the surface. And that's because we think Europa has a liquid ocean. There's a lot of evidence that suggests underneath that ice there's liquid. We're not quite sure what keeps that ocean warm and in a liquid state. We think maybe there's volcanic activity, so you have these underwater volcanoes that heat up the water and maintain it watery underneath. And it's also very salty. When you have this warm, salty material that can kind of float up through the ice is 7 kilometers thick. So it's a long way. It's protected. It's creating a protective shell between the surface of the planet where all that radiation is hitting and where life might be. But when you get this salty, warm brine, it floats up a bit like a lava lamp. And if you can imagine on the surface of that ice, that warm material blobbing up and poking through the ice and then freezing on the surface, you get a structure that looks like Europa. So we think Europa may have a liquid ocean. This is Enceladus. This is a beautiful moon. It has these tiger stripes. It's very famous for these tiger stripes. But what you notice about the moon is it's very smooth on this side, but pocked with craters on this side. And we think that this is, just like all moons, is getting bombarded with objects in the solar system. But the ice is actually turning over. It's smooth because once these impact craters form, the ice actually turns over. And they suspected that maybe there was liquid water under the surface of Enceladus that would explain why it was smooth. But we had no way of telling until the Cassini probe went past the moon and look back at it. And I don't know if you've ever seen people when they're talking to you and the sun is behind them and they have a few hairs sticking up off their head. You wouldn't normally see those hairs, but when the sun hits the hairs from behind and they stick up, you can see the hairs sticking up. Well, that's what happened with Enceladus. And this is the picture that the probe took, and you see these jets shooting up into space. And these jets shoot up about 200 kilometers. It's pretty spectacular. And the jets correspond with these cracks here. So what we believe is happening is that water in the subsurface underneath the ice is kind of sitting down on top of it, is creating pressure, and then that shooting the water up between the cracks in the ice out into the solar system. And this is, they've detected organic material in there. It's not unusual to detect organics. It's quite common in meteorites and comets. So there's organic material in water, which is also conducive to life. And this is the last place. This is the moon of Saturn. This is called Titan. And it's kind of smog gone crazy. So it's a dirty planet. It kind of looks a bit green. And it looks like it has a hydrocarbon problem. So they've not figured out how to control their smog. And the whole planet is coated in the smog, and it made it kind of hard to see through. It was just kind of a blob when you looked at it with microscopes. Cassini did a flyby, and it took these photos here, and you see these reflections. And this is sunlight reflecting off lakes on the planet's surface. Now, this is a very cold planet. It's about minus 170 degrees centigrade, and that makes it far too cold for there to be liquid water. But those hydrocarbons, things like ethanol and methanol, or methane even, when you cool them down enough, they turn into liquids. And this is actually a liquid of methane, so the same gas that would come out of a cooker or a stove when you're cooking. If you cool that down enough, you can actually turn it into a liquid, and you can make a liquid lake. So that is basically what they're seeing, is those hydrocarbons, this hydrocarbon smog is cooled down enough to the point where it makes liquid. The European Space Agency sent the probe down to the surface, and this is a photo that the probe took on the way down, and you can kind of see mountains and peninsulas here. You can even see deltas, almost like you see a delta on Earth where running water will carve out a river. But this is a lake of methane, and these rivers are formed by by methane flowing off these mountains and out into that methane ocean. So it's not liquid water we're looking at, it's liquid methane. But methane can behave much like water can if you're living in conditions, and you can survive conditions that get that cold. So now we've gone from an N of 2 to an N of 5 potential places that we can look for whether life can evolve in our solar system. And by understanding how life evolved on these planetary bodies, maybe understand how life could evolve elsewhere in the universe. So what do we know based on the information we have from planetary bodies in our own solar system? We know that there has to be a liquid solvent. So in most cases that needs to be water. But water is a solvent because it allows biomolecules, it allows our DNA and our proteins to float around and bounce into each other and interact. It doesn't have to be water that allows those interactions. It could be liquid methane, it could be liquid ethane, it could be anything that's a liquid. So that's why I say you have to have some kind of a liquid solvent. You have to have some kind of energy. And in all these places, because you're in the subsurface, you can actually use the energy of the rocks themselves. So you use the geological chemistry to drive the energy you need for life. And they're all protected in the subsurface. When you don't have that magnetosphere around you, that big magnetic balloon that protects you from the radiation of the sun, you need to hide underground to survive. So what does that look like? And this is something I can answer because I spend a lot of time underground. So, life in the subsurface, what do we know? I specialize in caves that are very deep and very isolated, and because of that, we don't have insects or invertebrates or other animals in there. is just driven by microbial activity. There is no photosynthetic energy. So most of the ecosystems that we understand on the surface where we live are driven by sunlight energy. Sunlight energy allows photosynthesis to fix carbon dioxide into plants that we consume, and our ecosystem is built on photosynthesis. This can't happen in the subsurface. There's no way for light to get in. So these communities live and grow, and the ecosystem functions in the absence of sunlight. They're physically isolated, so there is no interaction between these organisms and processes on the surface. Some of these sites that we study are 500 meters underground, and it takes about 10,000 years for any water on the surface to reach that area in the cave. And because of that, there's no real interaction between the two communities, and so we can study them in isolation. And they're also extremely starved. There's very, very little of the nutrients that you and I rely on to grow, to make our proteins, to make our DNA. So these organisms have to do it with all of those nutrients very, very limited, thousands of fold lower than you would see on the surface. And these are all conditions that you would find on these planetary bodies where we suspect life might evolve. And what will that life look like? It will probably look a bit like this. I'm sorry, this is a schematic, so this isn't actually what they look like. They're much more interesting than this, but they do kind of look a bit like sausages and soccer balls. The way that these are microorganisms, they're also much larger on this screen. They're very tiny. Most of the organisms I study, if you took 200 of them and put them head to tail, they wouldn't stretch the width of a human hair. It's very, very tiny. They grow and live using the same basic mechanism that you and I do. I'm sorry to get into physiology here, but this is my bread and butter. So they eat food. So imagine that you guys, do you have Snickers bar in Bulgaria? Yes, Snickers bar. This is a good one because everybody loves a Snickers bar. I'm not sponsored by them. Anyway. So you eat your Snickers bar, and your Snickers bar, even though it's tasty and it's chocolatey and peanutty, really what you're doing is consuming electrons. So the sugar in that Snickers bar is actually a molecule that is full of electrons. And those are the things that you need. So to obtain those electrons, you run it through something called the Tca cycle. It could be called the Krebs cycle. It's the central metabolic pathway. When you burn your Snickers bar in the Tca cycle, you push out carbon dioxide. And we know we do that because when we breathe out carbon dioxide. So when you're exhaling the off gases from your central metabolism that's burning that Snickers bar. It's also burning your chicken and your French fries and whatever you're eating. So you're burning it to release the electrons. And when you release the electrons, you have to pull them across an electron transport system that sucks the energy from that electron. And that energy gets trapped to do work in your body. Which is why if you eat a Snickers bar before you go for a 5-kilometer run, you do much better than if you don't eat your Snickers bar and try to run 5 kilometers. So, this is the basic process. You and I and everybody in this room, animals, most of our food is in the form of a carbohydrate, a sugar, or a protein. It all comes in this way. We're very much restricted into what we can burn. Bacteria are not. They can use organic material. They can take the waste product from a factory that is toxic to you and me, and they can break it down and burn it to release energy. They can take gases. They can take hydrogen sulfide gas. If you've ever gone to a sulfur spring and it smelled like disgusting eggs, that's a great energy source. They'll take that and they'll burn it. They can also eat minerals. So you hear about rock-eating microbes. They can actually take the mineral surface of a rock and break it down. Next time you look at one of these buildings and you see these lovely, beautiful old buildings in Sofia, and you see all those black patterns and coming down the surface, that's not all pollution. A lot of that is actually microorganisms living and growing on the surface of the buildings. So they can eat and burn those minerals. We use as an electron acceptor to pull those electrons out, we use oxygen. So the reason we breathe oxygen in is because it pulls the electrons through our electron transport chamber to trap the energy. And to show you how effective this is, we're going to put our hands over our mouths for 6 minutes. That's a joke. Okay. But if you do that, after 6 minutes, you've stopped producing enough energy that your brain will die. So this is how good your body is at converting this energy from food to do work to keep your brain alive. We have to use oxygen, and shutting off that oxygen to your body demonstrates how important it is to us. And we can't use anything else. You can't stop breathing oxygen and, I don't know, start sticking coal in your mouth. It's not going to work. We have to use oxygen. Microorganisms don't need to do that. They can use oxygen, they can use nitrate, they can use metals, they can even use CO2 gas. So all of these combinations here mean that these microorganisms are not trapped in the surface environment that you and I are, where we need sunlight to make the plants grow, the cow eats the plants, and then we have our steak. These microorganisms can breathe minerals, they can eat minerals, they can grow on surfaces. And this is how they make a living in the dark under extreme starvation. So this is one of our sample sites, and I always love to show it because you can see how cold it is on this photograph. No? No. Okay. It is minus 30 degrees centigrade when I took this photograph, and for some reason I decided to go here in January instead of in June, when it would have been about 25 degrees centigrade. It's very exciting to do microbiology when everything sticks to your fingers when you walk outside the car. This is where we were. This is a map of the United States, and this is South Dakota, and this is the Black Hills of South Dakota. And it's famous for Mount Rushmore, where they've carved heads of the American presidents. It's an interesting geology to it. It's a blister. Yellowstone, where all the hydrothermal systems and everything used to be under this area, but the whole landmass shifted north, so this used to be a lot like Yellowstone, and Yellowstone heated, the hydrothermal heat pushed up the mountains like a big blister, and then erosion sliced the top off, and it exposed this big band of limestone. And limestone is what caves like to form in. You guys have a big limestone mountain here behind Sophia. There's some nice big caves in it. So there are two national parks here. One is up here for Jewel Cave, and this is for Wind Cave. So this is Wind Cave National Park. It's one of the oldest national parks in the United States. It's over 100 years old. And it was created to preserve this. And this is a map of a cave. So you can see 200 meters for scale. It's about 1.5 kilometers on either side, and it's slightly over 220 kilometers long. So it's a very big, long cave. The natural entrance is up there, and it's a tiny hole. It's about 20 centimeters wide and maybe 25, 30 centimeters high. It's very, very tight. But luckily they put an elevator in so you can go in and go down. So the natural entrance is there. There's an elevator here, and then you can work your way through the cave down to this point here. It takes about 4 hours. If you want to get to this point here, it's a very fast team. It takes about 14 hours. So it's quite grueling to get through the cave. The reason I'm interested in this area here is because when I told you that there was a blister that kind of pushed the land up, when it pushed the land up, it tilted the rock. And so water from the surface kind of flows down. And it goes through the cave, and the cave tilts until eventually the cave intersects an aquifer. So there's a very large aquifer in this area. It's very ancient. It's up to 50,000 years old. It covers about a quarter of the United States. And the only way you can get to it without drilling is to walk in, through the elevator, drop down, and then drop another 200 meters through the cave until you hit the water table. And when you do, you come across a series of lakes. So these are not like lakes you would imagine where rainwater comes down and it's a stream, the stream pours into a lake and you have a big basin. These are lakes because the water is moving up and down underground, and as you walk down into the cave, you actually intersect where this groundwater has come up to create a lake. Now getting there is a bit of a challenge. As I said, it takes about 4 hours. It doesn't look that far. There's some of this. This is my postdoc. He's not wearing a shirt, not because he caved without a shirt on, but because it was so tight he couldn't actually fit wearing the shirt, so he had to take the shirt off to fit. He's actually the biggest person we've ever gotten through that crack. That crack is, I think it's less than 20 centimeters. Everything we take in the cave has to be smaller than 20 centimeters on its widest edge, otherwise it won't fit. And there's quite a bit of this, so everybody thinks my job is very glamorous until they actually come with me in a cave. And then you get to these lakes, these beautiful lakes. And when we got there, we found out that this is some of the cleanest water on Earth. Normally when you look at lake water, you might have 100,000 bacteria in every mil. And so even a lake that looks very clean is going to have bacteria in it. They're not going to harm you, they're part of the ecosystem, they keep the lake clean, they turn over contaminants. They're important to be in there. And 100,000 is about average for a clean lake. The ocean, when it looks a little bit cloudy, you might have maybe 7 million bacteria per mil, and you're drinking that, you're splashing around the ocean, it doesn't bother you. When we went into this lake, we were finding about 2,000 cells per mil. So it was 3 times cleaner than water that is considered clean. And it was very challenging to study because we have to get enough bacteria so we can study them by their DNA, which means that we need enough bacteria to extract DNA from. To collect enough material from this lake to extract DNA, we had to collect 300 liters of water. So we ended up filtering it in place to be able to extract the DNA. And that allowed us to collect about 500 nanograms of DNA. So to give you an idea of what 500 nanograms of DNA looks like, take a grain of sugar, cut it into 1,000 pieces, take one of those 1,000 pieces and cut it in half, and that's how much DNA we were able to collect, pumping 300 liters of water. The reason we did it is we wanted to look at diversity. So in, oh my goodness, this is fantastic there. You can see that's, there'll be a test on that list there at the end. We do phylum level. So when we think about life, you may have learned that there were 5 kingdoms on life on Earth. That's not strictly true. There's about 3 domains of life. The domain that we live in, the eukaryotes that have a nucleus, the domain that includes the bacteria, and the domain that includes the archaea. And the archaea actually look like bacteria, but are our cousins. So when we determined that there were these 3 domains of life as opposed to 5 kingdoms of life, it meant that the kingdoms that we thought about, animals, plants, fungi, were actually little branches within these domains. When you do that, there's over 300 kingdoms of life on Earth, and this is all done with genetic work, except we don't want to confuse people, we call them phyla. So this is kingdom-level distribution of bacteria found in that lake. So we're several hundred meters underground, we're extremely starved, there's no energy coming in, there's very few bacteria, and we have about 30 different kingdoms of bacteria within that lake, which was pretty fantastic. So even though it's super, super starved, it's super, super diverse, and everybody's working to make a living in that environment. Now, to collect it, we use this filter, and this is called a 0.2 micron filter. And the filter is right here, and we floated this thing in the lake, and it was important to use this filter because it allows you to filter out bacteria. So bacteria are quite small, 0.2 microns to make the holes in this membrane to filter bacteria. You take the membrane, and then you hit it with a neutrino, the gamma radiation. So they hit it with a radiation to make a little hole, and then the hole is expanded with a solvent to make a little gap. And that gap is much, much smaller than bacteria, about 5 times smaller than a bacteria. So when you float it and the water is filtered through the membrane, then you can collect the bacteria. When we took the water that filtered through, we thought this is super clean water, we'll take it back to the lab. And we added some nutrients to it, and something grew. It's like, there can't be anything growing in here. We collected all the bacteria. We took the culture where the bacteria grew and filtered them again, and they still grew. And it turned out the bacteria that we were isolating was small enough to get through this filter. So we went back into the cave with a filter which was half as big again as this, and we filtered the water again, and we found this group here. And 30% of the bacteria that we found within that cave are 5 times smaller than the usual size of the bacterium. So it turned out there was a remarkable level of diversity that no one was seeing because they were so small, we were losing them in the normal extraction techniques that we use to identify microbes. So, what do we know from the subsurface? What can we add to this? Liquid solvent, geochemical energy, protected in the subsurface, and they're also quite tiny. Now this is good for, this is Enceladus, where we think we have microbial life in the subsurface with these small levels of organics, because it provides some information on how microorganisms might be making a living in this environment. But the problem is, when you go to a regular cave that's made from limestone, is the geology doesn't match other planetary bodies. To have limestone on Earth, you have to have a lot of life. All of our limestone here is made by reefs, microorganisms living in the oceans. You don't have that on other planets. So the geology of a carbonate cave in South Dakota doesn't match the geology that we expect to see on a planetary body. So we decided to go after iron. And iron is important because iron is the dominant mineral on Mars. So, to study iron, we went to these mountains. These are mountains in Brazil. This is Belo Horizonte here. And these mountains are commercially important because they're made of iron. They're about 70% iron by weight. In fact, there's so much iron that what they do is they have these huge mines, and they will come in, they crush up the rock, they put it on a conveyor belt as a big slurry, they send it 300 kilometers to the coast as a slurry, it goes onto a boat, and it goes off to China and it's turned into steel. They don't even need to pre-process it. It's got so much iron in it, you can make steel to make buildings without having to process it. The interesting thing about these mountains is not only are they about one of the purest forms of iron that you can find naturally on Earth, but they're also full of caves. About 3,000 of them. So you have this small region with lots of iron, and it's full of caves. Now the issue is that iron isn't soluble. I mean, it will rust on your car. Eventually, if there's salt and water, you might have iron parts of your car that will rust away, but it takes a very long time. Iron itself is not really soluble in water. So the question is how could these caves form with water? So of course, I'm a microbiologist and a cave microbiologist, so I think everything is done by microbiology. We went into the caves and looked for the presence of microbes. When you go in them, you see things like this. This is something we call snotite or snotty. It looks like you've sneezed. It's all over the walls, and this is microbial. You can see all the microbes in here. The sediments, the floor was bubbling away, so something is producing that CO2 gas. You can see this dew hanging on the ceiling, so the whole caves were alive with microorganisms. To get to the cave is a bit of a challenge. This is my graduate student. This is just traveling to the cave. This is to keep the bees off, and then you have this suit and these gloves are for the bees, and then you have big boots to keep the snakes away, and then you have to have a guy that goes ahead of you with a machete. So it's you know, you feel like you're a real tough explorer, except they drive the car really close to the cave, so you dress up like this and you walk 80 meters and you're at the cave already. But you feel like you're doing the jungle, you know, Indiana Jones in the jungle. When we started sampling the caves, we found that the microbes were not on the surface in the caves, but when we drilled a hole into the rock, there was a gunge, like melting ice cream behind the rock. And it turns out that the microorganisms don't live on the surface, they live behind the rock itself. When you collect these microbes, You take some of the iron that the cave is made out of, mix it in a bottle, and then add the microbes. After a month, all the iron is reduced, it's turned into a liquid form. So these microbes are eating away at the cave. And they're doing it behind the walls. We put in this bottle here, we put just a chip of the iron that the cave's made out of. And this is the chip. We took a photograph of it before we put it in, and this is the photograph after, and you can see how the microbes have eaten away at the surface of the iron. So when we go into these caves, they are full of microorganisms, but the microorganisms are behind the rock. So what's happening is they're chewing away and eating at the rock, and then making it really, really unstable, and then whole pieces of the wall are collapsing and falling into the cave. So this is a cave that's forming not by water flowing through the middle of the cave, but by microbes eating it away and it collapsing in on itself, and you get this kind of bubble appearance. So, what has this told us about microbes in caves? Is they're tiny and they appear to do weird things. So if you don't understand what the microorganisms might be doing elsewhere, you have to be very, very careful in how you look for them. So when you ask the question of what does life look like, the last person you want to ask is a biologist. When we went into Wind Cave, we went with these filters because this is how you collect microorganisms, and we turned out to be wrong. When we went to Brazil, we went to look for microorganisms. We thought water has to flow through the caves, and microbes have to be living on the surface, but they were behind the rock. So you don't want to ask a biologist because we have very predetermined ideas of what life must look like. You see this, you say, yes, this is life. You see this, you say, yes, it's life. That's that N of 1. The idea of life is going to look like life looks on Earth. And there's no telling how life evolved on other planetary bodies. So when we do find evidence of planetary life, what will it actually look like? And it's probably going to look something like this. This was from a paper in Science from the Curiosity rover. And you see this peak here. You see there's a number 6 there, but it only goes up to number 5 there. That was evidence of a compound called chloramine. And chloramine is created by reacting methane with chlorine, which is found in the soils of Mars. And methane on Earth is primarily made by microorganisms. So this is it's not a smoking gun, it's not evidence of life, but it's evidence of the type of organic molecules that life makes potentially being present on Mars. So it's possible when we get that piece of data that everybody's been waiting for that suggests that there's life on Mars, it's going to look something like this, and your mother is going to ask you to explain what that means, so be prepared. And so the nice thing is, so this is Curiosity. We've managed to send rovers to multiple planetary bodies on our solar system, and we're going to be sending them out. And we've shown that these microorganisms can live under very starved conditions, including the conditions that you might actually find on the surface of the robot that you send, or the lander that you send. So we have to be very careful that we don't actually find life on Mars because we sent it by accident on Curiosity. And that's called Planetary Protection, to protect Mars from the invasive species that we accidentally drop off one of our rovers. Every instrument that goes to another planetary body, they look at the planetary body and it's ranked according to the possibility that life could survive, or there are conditions to support life once in 100 years. And if those conditions are met, then you have to be extremely careful to make sure your instruments are clean when you send them up. The other thing we noticed is that when we went into caves and we identified the microorganisms in caves and compared them to other microorganisms in the database would always come back with microorganisms from spacecraft assembly facilities. And it turns out all of the factors that allow microorganisms to survive that extreme starvation in caves also allows them to resist the extreme starvation that you might find on surfaces where you're building spacecraft. So I actually got to help on the Phoenix lander. This is really random. This could be a picture of anybody and I could say it's me, but you can tell it's me. Look, it's obvious. I thought it was really cool because I got to dress up. It's like, take my photo to Facebook. This is amazing. This is Phoenix in the background, and you can see there's these tubes going in here. And we're wearing these bunny suits, and we're all taped up. And they had these straps, and I thought that was like some flash to make it look really cool, you know, a bit like Star Trek where they have like the pins and the slashes. It's not. These are so it's easier to drag the body from the building if this blows up, because they were actually pumping in the hydrazine, which is the rocket fuel, while we were taking this photograph. So while I thought I looked kind of slick with my strap, it was actually so they could pull me from the building if it all went wrong. And I also, because of the work we've been doing in caves and trying to understand how we can recognize this very limited life under extreme nutrient starvation, we got very good at identifying contamination and how to control contamination. So Mars 2020 is a rover that's going to be sent to Mars to look for evidence of life. It's actually going to collect samples that can be returned to Earth. And one of the biggest considerations when you're going to collect a sample is how do you make sure that that sample you collected on Mars is not contaminated with life from Earth. This is that figure from the Curiosity rover again, and this is that peak that we're looking at, but this is the control. So this is just background, and this is all contamination. So our ability to control this level of contamination makes it more possible for us to be able to identify life if we find it on Mars. So one of the other big things that cave microbiology is being able to do is help us to understand how we can limit and control that contamination to make it more possible that we can identify life on Mars. So just to quickly, how did I end up standing on a stage in Bulgaria talking about astrobiology and working with NASA? If you'd asked me when I was a little kid what I was going to be, I would have probably said a vet. And then I volunteered at a vet's practice, and all the dogs and cats were throwing up all the time. It was disgusting. So anyway, I ended up going, well, I hate chemistry, and math is terrible, so I'm going to go biology, soft science, guaranteed win. So I started doing biology, and then I started my PhD in medical microbiology. And I started to understand bacteriology and microorganisms, how they function, how their genes work. And that took me into genetic regulation, so I started to understand genes, withdrawal, and virulence, and really got interested in bacteria. Understanding how these genes interact with each other, you actually have to do a lot of math, which is molecular phylogenetics, and it's these huge, long equations. But suddenly it made sense to me, because now it wasn't math because it was math. It was math that I needed to solve a problem, and that's kind of cool. I can do that. I can deal with that math. So I'm doing math now. Then I started looking at the geology and how the geologic environment influences the growth of the organisms. Like, geology is cool. It's rocks. I dig rocks, like fossils and things. I like that. But to understand how microorganisms change the surface of that rock, to be able to understand how they change its chemistry, and you can understand their biochemistry, I had to do materials chemistry. So by accident, I ended up doing math and chemistry anyway. And if you combine geology, materials chemistry, molecular genetics, genetic regulation, and biology, you end up being an astrobiologist. So it's just completely by chance, and if you look, it's not a straight line. And I think it's important to remember that if we study the things that interest us and we're not afraid of challenges, you'll actually get taken in a direction that is pretty cool, and you'll end up in a really cool place, Sophia. So I'd like to thank everybody who works for me. This is my lab in Akron. We're funded by the National Science Foundation, NASA, the National Park Service, National Institutes of Health, and the Fish and Wildlife Service. I think I have like 1 minute left, so I'm just going to, not everybody here is a caver. So caving is a pretty cool sport. When I tell students that come into my lab, I will teach you the ropes. I literally teach them ropes. So it does involve a lot of technical rope work. A lot of times we're doing that to avoid dangerous situations. We've had a couple of conversations about getting flooded in caves this trip. So this was just, we rigged this to bypass this waterfall. When we first found the cave, it it was completely dry, and then we're like, this will be fine, and then we're like, hang on a minute, what happens when it rains? This is a pit that we found in China. This was very cool. We threw a rock down, and we didn't hear it land, and we thought, oh, it must have landed in the mud, or got squishy or something. So we turned around and started walking away, and then we heard this boom. And this is what we found from up there. That's a person looking at a person. Can you see the body there? Yeah, well, actually you can see their light. It was a 421 meter drop to the room. It took 11 seconds for that rock to hit. It turned out that this room had, I think it was, I can't remember, it was over a 400 meter ceiling. It was 600 meters wide in its widest dimension. Turns out that volume-wise, it's the 4th biggest room in the world. It's not always big. Sometimes you're like this. Every caver will know that this isn't too bad because you don't have to take your helmet off. When you have to take your helmet off and blow all the air out your lungs so your lungs collapse, then you can say you're in a tight squeeze. And then of course it'll be like that with water. And then we're back again to floody cave type environments, which are a lot of fun. Anyway, caving is a pretty dynamic sport. I came into cave microbiology because I'm a caver. I was able to bring my sport to the science. And I think a lot of times we try to teach kids to keep sport and science or hobbies and science separate because they should be serious about one and the other's for fun. But it allows you to bring a lot of knowledge and a lot of technology to questions that other people can't answer because they don't understand your passion. So anyway, that's it. Thank you very much. Do I leave now? Yeah?